these in both biotic and abiotic stresses challenge the ability of the plant to perform well in adverse environmental conditions. So you see that plants generally exposed with wide range of abiotic stresses and these abiotic stresses generally affect growth, productivity, and these abiotic stresses, as we are discussing from the last 3 days, includes mainly drought, salinity, extreme temperatures, heavy metal toxicity, and nutrient deficiencies. And all these stresses basically disrupt physiological and metabolic process of the plants. But out of these stresses, particularly drought and salinity, are very detrimental and reduce photosynthesis and water use efficiency. So, on the other hand, if you see the biotic stresses results from the interaction with the living organisms such as pathogens, insects, nematodes, and weeds. So, generally these stresses generally compromise the immunity of the plant, leading to loss of yield. And the combined effect of biotic and abiotic stress, just I mentioned that it could be interactive, synergistic, and making it very fatal to the plant survival. So sometimes what happened that both plant face biotic as well as abiotic stress. So the combined effect could be additive, synergistic, making it very fatal to the survival of the plant. Now generally, as you see in this slide, that plant, they are exposed to number of abiotic stresses like heat, chilling, drought, waterlogging, salinity, and plants generally cope with these stresses by modulating activity of antioxidant enzymes, and these stresses also disrupt the activity of functioning of photosystem first, and second alter the activities of photosynthetic enzymes, reduces the stomatal conductance, and trigger loss of water. In addition to these stresses also cause hormonal imbalance. So this is also very pertinent that these stresses generally cause hormonal imbalance. Now I will just give you a brief overview how reactive oxygen species are produced in different cell organelles. Just I little bit elaborated in my previous lectures. So because also this hydrogen peroxide is very important as one of the antioxidant, so I will discuss it a little bit in more elaborate way. So you see that reactive oxygen species generally they are central to the plant responses to abiotic stresses. So what is the fate of this reactive oxygen species under normal conditions? So basically these are produced in a small amount as a byproduct of metabolic processes, and they play a very critical roles as a signaling molecule in growth and development. This is the fate under normal condition, but what happens during abiotic stresses, excessive production of reactive oxygen species disrupts cellular redox homeostasis, leading to oxidative stress that damages lipids, proteins, and nucleic acids. So you see, in one way it's produced as a normal byproduct and imparting signaling functions to regulate growth and development, but excessive production of reactive oxygen species leads to damage of lipids, protein, and nucleic acids. So how this reactive oxygen species is produced in chloroplast? So generally during abiotic stresses like high light intensity or during drought stress, there is an increased production of reactive oxygen species in chloroplast, and you see that excessive light energy generate generally singlet oxygen species and superoxide radicals in photosystem first and second, and there is a one important enzyme superoxide dismutase generally converts superoxide radical into hydrogen peroxide, which acts both as a signaling molecule and as a potential oxidant. So you see that hydrogen peroxide is uh, playing a dual role. In one way it is acting as a signaling molecule, in other way it is acting as a oxidant. So what is the fate of these reactive oxygen species production in mitochondria? So generally in response to abiotic stresses such as salinity, high temperature, generally ROs are produced in the mitochondrial electron transport chain, and mitochondrial production of reactive oxygen species, they are generally closely linked to energy metabolism and stress signaling pathways. What is the fate of these reactive oxygen species in peroxisomes? So during abiotic stresses like drought and high light, photorespiration in peroxisome is generally intensified, leading to increased production of hydrogen peroxide by enzyme glycolate oxidase. So peroxisomal production of reactive oxygen species are very critical for the metabolic adjustment during stress. So among the various reactive oxygen species, hydrogen peroxide basically plays a very central role due to its ability to diffuse across membrane. So this is very important point. You must focus on this, that hydrogen peroxide plays a central role due to its stability and ability to diffuse across membrane. Other molecules cannot easily diffuse the plasma membrane, but hydrogen peroxide can diffuse plasma membrane. So in this respect, hydrogen peroxide is very important as a signaling molecule. And you see that hydrogen peroxide acts as a double-edged sword. It serves as a, in one way it serves as a signaling molecule to activate defense mechanism, but can also cause oxidative damage if its concentration is not regulated. So this dual role of hydrogen peroxide makes a key mediator in plant responses to abiotic stresses. So by understanding this sort of connection, we generally we can explore strategies to balance hydrogen peroxide levels for improved stress tolerance in plants. So you see that concentration of hydrogen peroxide is very crucial. In one way it's acting as a signaling molecule, in other way it's acting as a oxidant. So it's acting as a double-edged sword. Now, hydrogen peroxide serves as a, generally a delicate balance is maintained between beneficial signaling and harmful oxidative stress in plants. So you see that from this slide you can have a view that there must be a balance should be maintained between its beneficial signaling and oxidative stress. So generally, as I mentioned, that at low concentration it acts as a signaling molecule regulating processes like stomatal closure, gene expression, activation of antioxidant defense system. However, excessive accumulation of hydrogen peroxide often due to abiotic stresses leads to oxidative damage to lipids, protein and DNA. So this dual nature of hydrogen peroxide makes it both as a protector and as well as a threat, depending upon its level and ability of the plants to maintain a redox homeostasis through efficient antioxidant system. So how hydrogen peroxide in plant is produced through both enzymatic and non-enzymatic pathways. And as I mentioned earlier also, that enzymatic sources include NADPH oxidase on the plasma membrane, which generate superoxide radicals that are converted to hydrogen peroxide by superoxide dismutase. Additionally, just I mentioned in my preceding slides that peroxisomes also contribute significantly via beta oxidation of fatty acid and photorespiration, where glycolate oxidase is a key enzyme responsible for this. Chloroplast and mitochondria also produce hydrogen peroxide as a byproduct of electron transport chain. In addition to this, non-enzymatic sources of hydrogen peroxide include spontaneous reactions such as dismutation of superoxide radicals in the apoplast, and the controlled generation and localization of hydrogen peroxide are very essential as it plays a dual role in signaling and oxidative stress depending upon its level and cellular environment. This makes hydrogen peroxide a versatile molecule in plant physiology and metabolism. Now, how hydrogen peroxide is detected by the plants, how it is perceived on the cell membrane. So, generally proteins present on the plasma membrane can detect reactive oxygen species like hydrogen peroxide and acts as a signaling intermediate by directly interacting with hydrogen peroxide. Although you see that hydrogen peroxide is very small, too small to have specific receptors, its oxidizing nature allows it to modify thiol residue present in the proteins, and extracellular hydrogen peroxide generated in the apoplast by enzymes like NADPH oxidases and superoxide dismutase. They are generally induced by various abiotic stresses which enters into the cytosol through aquaporins. Once hydrogen peroxide reaches inside, it oxidizes cytoplasmic proteins to regulate stress responses and growth. Recently, a major breakthrough in hydrogen peroxide signaling was uh, achieved by identification of HPCA1, which is a leucine-rich repeat receptor kinase located in the plasma membrane. And this HPCA1 is generally oxidized by hydrogen peroxide on the thiol group of cysteine residues in its extracellular domain. So this is a major breakthrough achieved in the signaling of hydrogen peroxide, and this oxidation basically activates autophosphorylation of HPCA1, which in turn triggers calcium channels and induces a stomatal closure. This discovery basically signifies and highlights the role of HPCA1 as a critical component of hydrogen peroxide perception and stress adaptation, offering a new insight into cell surface ROS sensing mechanism. Now, how hydrogen peroxide is transported? So, hydrogen peroxide transport across membrane is a very critical aspect of its role in signaling, but its polar nature poses a challenge for diffusion through hydrophobic membranes. To overcome this problem, hydrophobic nature of hydrogen peroxide, you see that hydrogen peroxide is transported via aquaporin channels, which facilitate its movement to regulate protein activity and signaling in the cytoplasm. And aquaporins homologs specifically from TIP and PIP families play vital role in selecting transport of hydrogen peroxide. In addition to these, a unique model of hydrogen peroxide transport in chloroplast have been proposed. I will discuss that. You see that the level of the effect of hydrogen peroxide varies significantly with its level ranging from beneficial as a signaling molecule at lower concentration, triggering a programmed cell death at higher concentration. So what happens at higher concentration of hydrogen peroxide? Generally oxidative stress is stimulated. So excessive presence of hydrogen peroxide leads to oxidative damage by inducing antioxidant system, causing lipid peroxidation, protein carbonylation, and DNA damage. Excessive concentration of hydrogen peroxide also damage DNA and repair. Generally, hydrogen peroxide induces DNA strands break and base modification. So this is a very crucial change brought about by hydrogen peroxide in DNA. If you see that repair mechanisms are insufficient, it can trigger cell death pathway. Now, programmed cell death is also one of the important repercussion of higher concentration of hydrogen peroxide. That high level of hydrogen peroxide levels basically activate the mitochondrial pathway of programmed cell death. So, what happens at lower concentration of the fate I described, what happens at higher concentration of hydrogen peroxide. Now I will discuss little bit what happens at lower concentration of hydrogen peroxide. Generally, it acts at a lower concentration, it acts as a signaling molecule and serves as a secondary messenger activating redox sensing proteins and signaling pathway. It modifies cysteine residues in redox sensors like thyroidoxin and glutaredoxin, leading to changes in their activity. Then at low concentration, it also alters gene expression. Activation of various transcription factors are well studied. It leads to closure of stomata. Basically, hydrogen peroxide signals guard cells to close stomata, reducing water loss during drought or high temperature. Basically, this involves sort of ion channel changes in ion channels and calcium signaling. Lower concentration of hydrogen peroxide is also very much crucial for cell wall reinforcement and also lipid peroxidation. Now, what is the role of hydrogen peroxide in photosynthesis? Just nutshell I would like to discuss. Basically, hydrogen peroxide at lower concentration protects photosynthetic apparatus of the plants during stress. It also regulates stomatal opening and optimizing uptake of carbon dioxide. In addition to this, you see that it mitigates, I am talking about the low concentration. So, low concentration of hydrogen peroxide basically mitigates photo inhibition under high light intensity. It also enhances repair mechanism for damaged proteins, promotes electron flow balance in photosystem 2 first and second. Now, you see this table reveals basically the impact of hydrogen peroxide on various photosynthetic traits in different crop plants like Zea maize, Vigna areata, Oryza sativa and tomato crops. So, if you want to have a quick look of the work done in relation to hydrogen peroxide, you can have from this slide. Now, what is the role of hydrogen peroxide in general in stress tolerance? So, generally a rise in hydrogen peroxide levels triggers several cellular responses, including modulation of intracellular calcium levels, which is very critical for activating protein kinase that initiate stress-related signaling pathways. So, as a signaling molecule, you see that hydrogen peroxide influences the activity of transcription factors in the nucleus. So, to counteract the potential harmful effect of excessive ROS production, basically plants have developed a hydrogen peroxide scavenging system that includes antioxidant enzymes like catalase, ascorbate peroxidase, as well as non-enzymatic antioxidants such as ascorbate and glutathione. So, these antioxidants basically works in concert to detoxify hydrogen peroxide and maintain cellular redox balance, preventing oxidative damage to lipids, protein and DNA. So these are basically fine, if there is any fine tuning in the regulation of hydrogen peroxide and its scavenging, uh, the plant's ability to cope with the stress will enhance, promoting resilience and survival under adverse environmental conditions. So, you see from this table describe the effect of hydrogen peroxide mediated abiotic stress tolerance in different crop plants. So, as a reference you can have a look of this table. Now, what is the role of hydrogen peroxide in drought stress? So, you see that hydrogen peroxide basically plays a very critical role in response of in plant responses to under drought stress by acting as a signaling molecule that mediates several adaptive processes. It induces stomatal closure, reducing water loss through transpiration. Uh, Basically, what hydrogen peroxide does basically, it activates drought responsive gene pathway, enhancing its production of protective metabolites that helps to mitigate cellular damage. Additionally, hydrogen peroxide also regulates antioxidant enzyme activities, maintaining a redox homeostasis under stress condition. Furthermore, hydrogen peroxide interacts with also one of the important growth hormone, that is abscisic acid, uh, which is responsible in drought signaling uh, to coordinate responses of plants that optimize water conservation and cellular protection. Now you see this figure basically demonstrate that uh, S2 treatment could help to mitigate drought induced morphological damage in soybean plants. So plants under severe drought condition with application of hydrogen peroxide show better stress tolerance compared to untreated severe drought stress plants, highlighting the protective role of hydrogen peroxide in drought stress. So you see that this figure shows the effect of drought stress on soybean plants on various physiological parameters with or without application of hydrogen peroxide. Generally, various parameters were altered by application of hydrogen peroxide like relative water content, photosynthetic rate, stomatal conductance, and transpiration rate of soybean plants. So you see that how hydrogen peroxide is modulating very significant metabolic parameters of the plant. Now, what is the role of hydrogen peroxide in salinity stress? So, you see that hydrogen peroxide also plays a very critical and vital role in enhancing plant resilience to salinity stress by serving as a sort of signaling molecule. You see that it regulates ion homeostasis, helping to maintain a balance between sodium and potassium ions, which is very critical for cellular functions under salt stress. Hydrogen, in addition to this, hydrogen peroxide also mitigate oxidative damage by scavenging excessive production of reactive oxygen species through activation of antioxidant defense mechanism, including enzymes like superoxide dismutase, catalase. And in addition to this, you see that in one way it is inducing the antioxidant enzyme, it also promotes the synthesis of osmoprotectant. This is also very crucial, such as proline and glycine betaine, which generally stabilizes cellular structures and maintain a sort of osmotic balance. Additionally, hydrogen peroxide also interacts with hormonal and signaling pathways, including abscisic acid and nitric oxide, to activate salt tolerance gene and stress responsive pathways. So these multifaceted roles underscores the importance of hydrogen peroxide in plants against the detrimental effects of salinity stress while supporting growth and metabolic functions. Now this slide shows a simple graphic depiction of the main molecular mechanism in response to plants treated with hydrogen peroxide and how they could be improved salinity stress tolerance in plants. So this table basically provides information about the concentration and timing of hydrogen peroxide priming in plants along with its positive impacts and mechanism in reducing the salinity stress. It highlights how optimal level of hydrogen peroxide at the right time improve the ion balance, enhance antioxidant defense and promote osmoprotectant production and activate stress responsive pathways, all of which is helped plants to adapt to and withstand the harmful effect of salinity stress. So from these findings, you just have a look that how hydrogen peroxide is mitigating various stresses, particularly salinity stress. Now, what is the role of hydrogen peroxide in temperature stress? Uh, why I am elaborating all these role of hydrogen peroxide in all these stresses? Because this is a very versatile molecule, and you see that its use could be implemented in any sort of abiotic stresses. That's why just I am giving a brief overview of all these stresses in nutshell. So in addition, in continuation, what is the role of hydrogen peroxide in temperature stress? You see that hydrogen peroxide basically modulate heat shock proteins during high temperature, and hydrogen peroxide also is very crucial for protecting photosynthetic machinery from damage to heat stress, and it also regulates cold responsive genes under low temperature, and in general it enhances antioxidant enzyme activities, and thereby it aids in maintaining cellular stability under extreme temperature. So this is the role of hydrogen peroxide uh, in temperature stress, just I discussed in nutshell. Now this is the model that how S2O signaling cascade is triggered under low temperature stress. This is a very relevant figure. If you go through the figure, you see that under low temperature stress, what happened? Hydrogen peroxide acts as a signaling molecule and initiating various defense responses in plants. It triggers an increase in the intracellular calcium levels, which acts as a secondary messenger to activate downstream pathways. The mitogen activated protein kinase cascade basically is then activated, amplifying the hydrogen peroxide signals, where you see that S2O2 hydrogen peroxide also interacts with glutathione pool and converting reduced glutathione to its oxidized form, then which modulates redox homeostasis. So this redox signaling in turn activates transcription factors, and these various genes encode proteins which enhance cold tolerance by stabilizing cellular structures and maintaining metabolic functions. So in addition to this, hydrogen peroxide under cold stress condition modulates activity of catalase, balancing the production of ROS level to prevent oxidative damage while ensuring effective signaling at the same time. So through these intricate networks, hydrogen peroxide orchestrates, or you can say involved in plant responses to low temperature stress, enhancing the survival of the plants and its further adapted mechanism. Now this is the figure just my student published one of the review paper on hydrogen peroxide. So in nutshell, what is the role of hydrogen peroxide in heavy metal stress? We can have a quick look that the generation of reactive oxygen species including hydrogen peroxide activates a complex signaling network that helps plants to respond and adapt under heavy metal stress. Basically, as I mentioned that hydrogen peroxide acts as a signaling molecule by interacting with specific receptors and redox sensors such as calcium channels and cysteine kinases, which initiate a cascade of intracellular events. And this includes the activation of the mitogen activated protein kinase pathway, amplifying the stress signals. So, basically hydrogen peroxide modulates the redox status of the cell, interacting with antioxidants like ascorbate and glutathione, regulating oxidative stress and preventing cellular damage. So, these interactions along with the formation of various regulatory proteins lead to the activation of transcription factors which promote the expression of genes responsible for metal detoxification, antioxidant defense and cellular repairs. So, this is a model showing how hydrogen peroxide could protect plants under heavy metal stress. In addition to its direct role, hydrogen peroxide interacts with other plant growth regulators such as abscisic acid, auxins, cytokinins. For example, you see that hydrogen peroxide enhances the signaling of abscisic acid, which helps to manage loss of water and promotes stress tolerance under drought stress. Then hydrogen peroxide also crosstalk with auxins, cytokinins to modulate growth and development under abiotic stresses. So, basically these interactions along with the hormone, other hormone, fine tune the stress response of the plant, balancing growth and defense mechanism. Therefore, hydrogen peroxide works along with other plant hormone and signaling molecules to just modulate the multifaceted response of plants to heavy metal stress, improving tolerance and survival of the plant. So, this is a very crucial figure. Now, this is the table showing the emulative role of hydrogen peroxide in heavy metal stress. So, if you want to have a quick look that how hydrogen peroxide could be applied and at what concentration it should be applied to the plants to generate positive response. So, from this table you can have a quick look. Now, just see that how hydrogen peroxide interacts with multiple phytohormones, creating a complex network of signaling pathways that regulate key plant functions. This crosstalk influences processes like formation of fruit, germination of seed, stress tolerance and cellular response to abiotic stresses. So, if I talk about what is the role of hydrogen peroxide in formation of fruit, basically hydrogen peroxide interacts with auxin to regulate initiation of fruit, whereas auxin promoting production of hydrogen peroxide, which in turn facilitates the formation of root. So, how hydrogen peroxide interact with gibberellins? So, hydrogen peroxide basically modulates biosynthesis of gibberellic acid by influencing enzymes involved in production of gibberellic acid, promoting seedling growth and under favorable and under unfavorable conditions. So, hydrogen peroxide also reacts with cytokinin, it also reacts with brasnosteroid, and Andres has already discussed detail the role of brasnosteroid under abiotic stress, so I am not going to repeat it. And hydrogen peroxide also interacts with jasmonic acid and activates antioxidant system. So, you see that hydrogen peroxide at low concentration does not act in solo. It also interacts with other plant hormones. The motive to show this slide is that hydrogen peroxide does not act in solo, it interacts with other plant hormones to carry out number of specific plant growth and developmental functions. Now, moving ahead, you see that hydrogen peroxide also interacts with other hormones like nitric oxide, Yesterday, Dr. Gauzia Bashiri discussed a detail regarding the role of nitric oxide. So, my point is that hydrogen peroxide also interact with nitric oxide and calcium ion, and which are very crucial signaling molecule in plants that regulate physiological processes, development and various stress responses. You see that various abiotic stresses trigger the production of hydrogen peroxide, nitric oxide and calcium, and hormonal signals like abscisic acid can also act as a stimuli, and basically nitric oxide enhances activity of NADPH oxidase, leading to increased production of hydrogen peroxide, and then together they regulate calcium signaling, stomatal movement and transcription factor activation. Hydrogen peroxide also modulates calcium influx by opening calcium channels present on the plasma membrane. This leads to a spike in the cytosolic level of calcium, which acts as a secondary messenger. So, in addition to this, mitogen activated protein kinases are very central to integrating signals from hydrogen peroxide, nitric oxide and calcium. These mitogen activated protein kinases basically transduce signals to regulate gene expression, activation of enzyme and production of defense molecule. So, these cascades basically influence the plant growth like root elongation, leaf expansion and various stress responses like production of osmolytes and activation of antioxidant defense system. So, this is the figure showing the retracted from one of my published paper in Chemosphere this year in 2025, and this figure talk about a crosstalk of hydrogen peroxide with polyamines and nitric oxide and calcium during abiotic stress, and how mitogen activated protein cascade is responsible for activating various responses in plants. So, various abiotic stresses generally could trigger the level of polyamines, just I discussed in detail in my previous lecture. An elevated level of polyamines and calcium basically promote elongation of root, enhancing tolerance to abiotic stresses such as drought and salinity. So, this rise in the level of polyamines basically stimulates one of the important crucial enzyme, polyamine oxidase, leading to generation of hydrogen peroxide. So, here hydrogen peroxide come into play by increasing the level of polyamines. So, this sort of interaction with polyamines, hydrogen peroxide and calcium activates various reactive oxygen species signaling pathways and increases activity of mitogen activated protein kinase. So, this in turn basically induces the production of transcription factors that regulate the expression of stress tolerance genes, thereby enhancing the resilience of plants to various abiotic stresses. Now, I will just in a quick, I will show you some of my case study dealing with the interactive role of epibresnolone and hydrogen peroxide in regulating stomatal physiology, root morphology, photosynthesis and growth rates in Solanum lycopersicum under nickel stress. So, this slide shows the effect of various treatment of hydrogen peroxide, one of the bresnolone in combination to nickel stress plants, and how these hydrogen peroxide, epibresnolone in solo or in combination affected the growth of the plant grown under nickel stress. So, you see that how the plant morphology, you can visualize it change with those plants which were growing under nickel stress, and then application of low concentration of hydrogen peroxide and epibresnolone mitigated the damaging impact of nickel. Now, so you see that if plants are grown in any sort of heavy metal stress, it is going to damage the morphology of the root. So in this case also the morphology of root was severely affected by the plants supplied with nickel. So once the plants were given treatment of low concentration of hydrogen peroxide, it generally restored the morphology of the root, and it is very much clear from this slide. Now this figure shows how the photosynthetic parameters were altered by nickel, and you see that low concentration of hydrogen peroxide was very effective to overcome the nickel stress. You see in this slide that how Bresnoy's triad and hydrogen peroxide help to mitigate the nickel-induced changes in antioxidant parameters. Now this is another case study from my lab where role of hydrogen peroxide in modulating photosynthesis and antioxidant defense system in tomato berry studies where plants were given copper stress. In my previous studies it was nickel stress, here it is copper stress. Now you see that how stomatal responses were altered by plants given with nickel stress. The high concentration of copper led to closure of stomata, but once low concentration of hydrogen peroxide were given to the tomato plants, it restored the normal morphology of stomata. Now one of the interesting methodology I would like to discuss what is the role of diaminobenzidine and nitrobutazolium, and these two chemicals basically they are widely used as a tool to detect reactive oxygen species in stress plants. So these two dyes, basically diaminobenzidine and nitrobutazolium, they are used as a tool to detect reactive oxygen species in stress plants. So their basically mechanism is based on their ability to visualize oxidative stress by staining specific reactive oxygen species such as hydrogen peroxide and superoxide radicals. So basically this diaminobenzidine specifically detects accumulation of hydrogen peroxide. So in this context this diab dye is very significant. It can detect and bind the hydrogen peroxide. It basically reacts with hydrogen peroxide in the presence of plant peroxidases to form a reddish brown polymer which appears as a distinct stain on the plant tissues, and these reactions highlight the regions of H2O2 production often corresponding to areas under abiotic stress such as drought, salinity and heavy metal stress. So likewise if I talk about another dye, nitrobutazolium, basically this dye detects superoxide dismutase radical by reacting with the superoxide to form formazan, and this formazan as insoluble blue compound. This blue color marks region of the superoxide accumulation which often occurs in organelles like mitochondria, chloroplast, plasma membrane under stress condition. So these staining methods are basically very much instrumental in students carrying out research on abiotic stresses, as their visualization and localization of ROS in plant tissues is very crucial. Just I mentioned that DAB staining is very crucial for visualizing the accumulation of hydrogen peroxide, whereas NBT is very significant for visualizing the superoxide radicals. So just to have a look of how these two dyes they are significant and playing a role to detect superoxide radical as well as hydrogen peroxide. That's why I discussed it here. Now you see that how these accumulation of hydrogen peroxide and superoxide radicals was detected. This slide clearly shows it. Now I would like to conclude my lecture with that hydrogen peroxide exhibits a dual nature in plant cell. At high concentration it causes oxidative damage, uh, and like lipids, protein and DNA, potentially leading to cell death. However, at low concentration, hydrogen peroxide functions as an important signaling molecule involved in regulating plant responses to various abiotic stresses. In addition to this, hydrogen peroxide also plays a significant role in plant responses to abiotic stresses, and it participates in systematic signaling and may regulate various physiological processes, helping plants to adapt to various environmental challenges. Hydrogen peroxide signaling is basically closely integrated with other signaling networks in plants, allowing for the coordinated regulation of developmental processes and stress responses. So this integration ensures that plants can efficiently manage their internal responses to changing environmental conditions. Now, what are the future prospects? The exact site of hydrogen peroxide production under different stress conditions are still a matter of debate. The identification of dedicated sensors or signal transducers complex for hydrogen peroxide related proteins is still lacking in plants. The direct role of hydrogen peroxide as an activator in stress responses remains still not clear. Understanding the correlation between hydrogen peroxide oscillations and other signaling pathways will be a major research focus. Additionally, determining the specificity of hydrogen peroxide and how it interacts with other stress-related pathways will remain a key challenge for plant biologists in coming years. Thank you for very much for listening my lecture. If you have any query, just you are free to ask and raise.